Hey everyone, welcome again in this topic. So in the previous video, we were discussing very important concepts that you have to know in this topic of genetics and inheritance. So those terminologies are very important because of you need to know them before you can commence to solving problems involving genetic crosses. So in this video, I'm going to come up with examples from my mind because of I didn't prepare examples so that I can be able to do them here to show you how the courses are done. So I'm just going to use my mind to create scenarios based on the genetic crosses and then try to solve those problems and see and show you how those crosses are done, and then after that you'll be able to complete any question that you're going to do. So in this video I'm going to start with simple monohybrid crosses problems. So those problems are going to include the inheritance of sex, how one is able to become a male and a female, and then also the inheritance of blood groups, how I'm able to have blood type A, B, or O. And then before I can do those ones, firstly I'm going to show you how maybe some characteristics that are not sex linked are able to be acquired. Maybe the color, a fair color in animals, how the color is able to be acquired, and also maybe try to show you how the color in plants are able to be inherited. Maybe when you check the flowers, some flowers are yellow, others are red, and then others are white. So I'm just going to do maybe a quick example about maybe a black flower and then a white flower from my mind. So first of all, we have to know what is a monohybrid cross. In the previous video, I told you guys that a monohybrid cross is a type of cross whereby we focus in the inheritance of only one characteristic, or whereby we cross two organisms. When we cross them, we only focus in one characteristic that we're going to study. It can be height, or the characteristic can be color. So this is a monohybrid cross. So there's a template that we have to know, that we have to follow at all the times when we do these courses. So first of all, I'm going to show you that template. I'm calling it a template, but then it is not a template, but then that's how I call it so that you can be able to understand it better. So we're going to use these inverted commas and call it a template. So first of all, always when you use this template, you're going to start with the first parental generation, P1 generation. So in the P1 generation, the first thing that we're going to focus in is the phenotype. So in the previous video, we said that a phenotype is what you see in the outside, that is the physical appearance of an organism, right? So the phenotype, because of it, is what you see. In our case here, we're going to say that a black flower, it is crossed with a white flower. So let's say it's a black flower and then crossed with a white flower. So here our phenotype is what? It is black for the first flower. For this first flower, the black flower, we're going to say that the phenotype of this flower, because of it, is what you can see on the outside of the physical appearance of this flower. It's black. So we're going to say that the phenotype, it is black. A black flower, it is interbreeded, or it is crossed with a white flower. So phenotype of another color, of another flower, it is white. So a black flower is crossed with a white flower. After that, now we're going to check our genotype. How are the genes of these flowers look like? The genotypes. So the genotype. So let's say that our flower, this one, it was homozygous black, and then black is dominant over white, as you can see here. And it is very important for you guys to note that we are not using different alphabets simply because of this. We're talking about different colors. For example, you can see that we said black, then we use B. When we come to white, we're not saying W. We use B again. Why? Because of these are the same alleles. They code for the same characteristic, which is color. So we have to use the same alleles. But then to differentiate them, to show that the other one is dominant and then the other one is recessive, we're using different capital letters. We're using capital letter B for dominant to show that B, it is dominant or it is above white. That's why we represent it using the small letter B. So then I said that, let's say that this black flower, it was homozygous. It was homozygous, meaning it had the same alleles for black. It was homozygous black. So if it is homozygous black, it means that it had this genotype. It has capital letter B's for black, and then remember that black is dominant over white. And then remember that a dominant allele is an allele that is going to influence the phenotype, the physical appearance. This one is going to influence the physical appearance, meaning if we are going to have this thing at the end, that means that automatically our offspring will be black. And the recessive allele, this one is small. You can see that this one is able to mask it. So it means that the recessive allele, in the previous video, I said that the recessive allele is the allele that can only influence, or that can only show up in the phenotype when it is in a homozygous condition. Only if we have the two small B's together, that's when it can only influence the phenotype. I hope you guys understand. And then it's crossed with a white. Let's say this one, it is also homozygous. It's homozygous white, homozygous black, meaning that it's going to have the small letter B's like this. So this it is the parents. These are our parents. That's why we call them the first parental generation. So these parents are crossed together or are interbreeded together so that they can have offsprings. So you wanna see how those offsprings look like. So we all know that before these parents can have offsprings, they have to undergo meiosis. We did the topic of meiosis in the previous topic, right? So in the previous topic we said that the meiosis is going to form the gametes. That's why we're going to include it here. So now these parents, they undergo the process of meiosis. After they undergo meiosis, the meiosis is going to form the gametes. So when meiosis comes here, because we have a diploid parent and then also a diploid parent, these cells are diploid. So meiosis is going to have the number of chromosomes. You all know that it's a reduction division. So meiosis is going to form two possible gametes. It can form this gamete. It can also form this gamete. So I'm using a comma here to show that. I'm using a comma here to show that these gametes were formed from here. It means that our diploid cell is split from haploid gametes. And then the same thing is going to happen here. Meiosis is going to form two possible gametes. Right? So since now we have the gametes, that means that these gametes, they have to undergo the process of fertilization. So now these two parents, they have to interbreed now. So when they interbreed, therefore the process of fertilization is going to take place. So you're going to experience fertilization. So fertilization, I'm going to show you using what we call a Punnett square. So this box is called a Punnett square. So it is used in genetic crosses to show how the gametes are able to fuse together during fertilization. So I'm going to use this Punnett square. Okay, so in my Punnett square, I'm going to start with these two gametes for black parent that were formed by a black parent or a black flower. I'm going to put them at the top here. I'm going to bring this B here, and then this B here, and then these two, I'm going to bring them here, small b's. So after that, I'm going to show how these gametes are able to fuse together. So this one multiplied by this one, they're going to be BB. It's very important for you to always start with the capital letter B. So this one combined with this one, going to say it's going to form BB. This one multiplied by this one, going to be BB again. After that, this one has to combine with this one, and it forms BB again. And then this one has to combine with this one, it forms BB again. So you can see that now these gametes, after they fuse, they form these possible offsprings. These are our possible offsprings. So it means that we are going to have the first generation. We call it the first filial generation, but you can just call it F1 generation. So in our first filial generation, we're going to see the genotype first, because it is what we can see with our eyes when we look at this whiteboard. We see the genotype, not the phenotype. We're not able to see is it black or brown or what. So we're only able to see the genotype. So the first filial generation, or the first generation in short, we see its genotype. So as you can see, all of them are capital letter B, small letter b. So the genotype, we're going to say all of them are capital letter B, small letter b. After that, you can deduce the phenotype from what you see, because of, remember at the start of the question we said that B is for black and black is dominant over white. So it means that in this thing we have a heterozygous condition, but then we have this dominant allele that is going to tell us what is the phenotype. Okay, so the phenotype now, we can say all of them will be black. When we look at the offsprings at the end of the fertilization period, we see our offsprings becoming black. Why are they black? Because of this dominant allele which represents black, and it is dominant. So once it is dominant, this allele, it is going to influence the phenotype. It's going to show us what is the phenotype because of it is dominant. That's why we are able to have black offsprings at the end. All of them are going to be black because of all of them they have capital letter B, small letter b. So it means that we have what you call a heterozygous condition because we have different alleles in our case. I hope you understand. So it is very, very important for you guys to be able to see, to be able to use this template. You have to use this template wisely because of each and every aspect in this template has its own mark. So once you know this template, you're going to be able to solve any problem that we are going to encounter when you solve the genetic problems. I hope you understand. So now I'm going to try to cross the offspring that are heterozygous and show you the possible offsprings that can be formed by the parents if the parents have this phenotype or genotype. Okay, let's say now we have two flowers. Let's say we have two flowers, and then these 2 flowers, all of them are heterozygous for black color, meaning all of them are black, meaning they have this phenotype. So all of them, they have this genotype. And then obviously if they are like this, it means that they are black in color. They are black in color. So this is the same example, but then now I'm crossing the parents if they are like this. So it's the same example. So you want to know if you have the parents that are black, but then they are not homozygous like these ones. You see this one were homozygous, but then now we want to see if they are heterozygous, they have small letter b, and small letter b, capital letter b. How are the offsprings at the end of the fertilization period? So I'm going to use the same template. I'm just going to substitute in my template to try and see how are the parents. How are the offsprings at the end of fertilization? So first of all, I'm going to start with the phenotype of the first generation. The phenotype is going to be black. A black parent flower is caused with another black flower, because of they are black. This is the phenotype. I'm here in this step. Now I'm here in the genotype. What is the genotype? Here is the genotype. It is B crossed with B is the genotype. Then after genotype, what comes? It is meiosis. So now meiosis is going to occur. Meiosis. Why does meiosis occur? It forms the gametes. So it means that meiosis has to form the gametes. So now meiosis is able to form 2 types of gametes. It can form the one with capital letter B and then with small letter b. And then this side meiosis can form the same thing, capital letter B, small letter b, the gametes. After that, these gametes now have to fuse. Then they have to undergo the process of fertilization. And I said that during fertilization, these gametes have to fuse. So I'm going to use a Punnett square to show how these gametes are able to fuse. Now I'm going to use a Punnett square. So I'm going to start with these ones. I'm going to put them at the top, and then this one at the side. So now when they fuse during fertilization, when they combine the gametes, this one can combine with this one to form capital letter B's, and then this one can fuse with this one to form capital letter B, small letter b. Okay, and then this one can fuse with this one to form capital letter B, small letter b, and then this one can fuse with this one to form small letter b's. So you can see that now after fertilization, the genotype of our offsprings, they are not the same, they are different. We only have 2 of them that are the same, which is this one and this one, because they have capital letter B, small letter b, capital letter B, small letter b. So after this step now, we're going to have this step of F1 generation. I'm going to write it here. So now we're going to have the first filial generation, and then we're going to have the genotype first of that generation. In our genotype, you can see that we have 1 Ilorenki BB, and then we also have another one which is BB and BB. So 2 of them are BB and BB. So you can see that 2 of them are BB, to the letter B, small letter b, and then lastly we have 1, this one with small letter b's. So you can see that the ratio here in our case is 1:2:1. Because of 1, they are the first one we're going to have an offspring that is going to be homozygous black. You can see that it has the same genotype. The alleles are the same. This one it is heterozygous. You can see that the alleles are different. And then this one it is homozygous again. Okay, so this one it is our genotype. And in our phenotype, this is what we're going to say. Phenotype. So in our phenotype, we're going to say that we have 1, 2, 3. It's 1, 2, 3 because the 2 plus 1 is 3. So we're going to say that we have 3 of them that are black. So it means that 3 offsprings can become black. Why 3 of them can become black? Because of, you can see that we have this capital letter B, the dominant B, which shows us that B is for black. It's dominant. It's going to influence the phenotype. That's why wherever we have capital letter B, we're going to say that the phenotype is going to be black. That's why I'm saying 3 of them are black. Then here we have the small letter b's. It means that this one it is white. Why? Because of it has a homozygous condition with recessive alleles only present. And then the recessive alleles here, they show us a white color. So it means that only one can become white. Only one can become white. Okay, so I hope you guys have understood what I have done here. I want you guys to watch this video regularly until you understand this template, and then until you understand the whole procedure of doing these crosses. Okay, I'm going to see you guys in the next video, whereby I'll be shooting about the inheritance of sex to show you how are you able to become a male or female, and then lastly I'm going to show you guys how are you able to inherit blood group from your parents. Thank you.